Hi there, my name is Cora and I'll be talking a little bit about epistatic pathways. To start, epistasis is the result of interactions of alleles at different loci that cause the expression of alleles at one locus to mask or change the expression of alleles at a different locus. The gene that does the modification and is expressed is known as epistatic, while the other is the masked gene. Because of epistasis, several genotypes can result in the same phenotype. For example, this is a model of a single recessive epistatic pathway using the fictitious example of coat color in unicorns. The first locus blue is dominant to yellow, which would be considered the colorless option in real organisms. The second locus, rainbow, is dominant to black. If we do a traditional dihybrid cross, we can follow the pathways to determine the expected phenotypic proportions and compare them to Mendelian proportions of 9, 3, 3, and 1. For offspring possessing a dominant allele in both loci, the dominant allele B will activate enzyme B for the creation of blue, and then the dominant allele R will allow for enzyme R to create rainbow coat color. Out of 16 organisms, 9 will have a rainbow phenotype. For offspring with the first locus dominant and the second recessive, the dominant B will allow for enzyme B to create blue. However, there will not be an inactivation of enzyme R. This will result in 3 of the offspring having a blue coat color. For the first locus recessive and the second dominant, the enzyme B will not be activated, so it does not matter what the R allele is, ending in the coat color as yellow for 3 of the 16 offspring. Finally, with both loci recessive, there is also no enzyme activation, resulting in 1 additional yellow coat color. Overall, this results in 9 unicorns with a rainbow coat color, 3 with blue, and 4 with yellow. This helps to show that in some traits where the first locus has homozygous recessive alleles, it will be epistatic to the second locus, causing only the expression of the homozygous recessive alleles. In an example that is not a typical dihybrid cross, we will cross a blue unicorn and a yellow unicorn with the genotypes listed on the screen. We determine the genotypes of the offspring probabilities using a branch diagram. The blue unicorn has a 50% chance of passing on either a dominant or recessive B, while the yellow unicorn can only donate a recessive B. At the second locus, the blue unicorn can only donate a recessive R, and the yellow has a 50% chance of passing either the dominant or the recessive R. From these genotypes, we can follow the pathway like we did in the dihybrid cross. For 1/4 of the offspring, the pathway can be completed and the coat color will be rainbow. For 1/4 of the offspring, the pathway can only be halfway completed, so the coat color will be blue. The remaining 2/4 of the offspring possess homozygous recessive alleles in the first locus, so the pathway cannot be started, resulting in yellow coat color. Overall, out of 16 offspring, we would expect 4 to be rainbow, 4 to be blue, and 8 to be yellow.